Hello, everyone. I see some people joining. We're just waiting a few minutes, and then we'll get this webinar started. All right, guys, I think it's officially time. So it is my sincere pleasure to wish you all a warm welcome to this webinar with the title, Unlocking the Spatial Dimension, Maximizing Insights from Spatial Omics Data, which is co-hosted between BioLizard and PixelGen Technologies. And my name is Annika Branting, and I'm the CCO at PixelGen, and I will guide you through today's presentations. And please feel free to use the Q&A function to leave questions for the presenters, and we will address them at the end of each presentation. So in this first section of this webinar, we will hear from Louise Leonanker, FAS at PixelGen, who will talk about a new capability in spatial analysis, namely being able to investigate the cell membrane protein dynamics of single cells in solution. So for the next 20 minutes or so, we will focus on cells in solution rather than tissue, while Louise explained why it's important to understand the spatial relationship between proteins on the cell surface and how you can obtain that type of data. So Louise, please go ahead.
Yeah, thank you so much for the introduction, Annika. So I really look forward to guiding you through molecular pixelation and how that can be used for novel insights into biology. So first off, I want to talk a little bit about the spatial field. So spatial techniques are typically thought of in the context of tissue samples and using some sort of imaging. Today, I want to switch gears a bit and talk about spatial in the context of cells in suspension and how you can achieve spatial protein information without the use of imaging, but instead with a digital output. So that is what molecular pixelation does. So molecular pixelation is the technology that we have developed here at PixelGen, and we are pioneering spatial proteomics for single cells. So why is it so important to understand the cell surface proteins, and how can it be so closely linked to cellular function? So we have some examples here where the first one is communication. And you can see here how a T cell and an antigen presenting cell, how they're interacting through the so-called immune synapse, which is a very specific protein constellation on the cell surface that makes it possible for them to communicate. Another example would be an action where a CAR T cell is interacting with a cancer cell, and it needs to reorganize some of its cell surface proteins in order to bind and perform this perforin granzyme-based cancer cell killing. And a third example would be attachment and movement, or migration, and how migrating T cells also reorganize their proteins to this back part of the cell as it's migrating, something called a uropod, which makes the cell more stable as it needs to migrate into packed tumor microenvironments to perform cytotoxicity. We also have the fourth example of a mode of action and an antibody drug. So what you see here in pink is rituximab that binds to CD20 on B-cell cancers, and that clusters them or polarizes them to one end, which makes it possible for NK cells to bind, and NK cells will then initiate the ADCC pathway that ultimately leads to cancer cell killing. So those are some examples of where it's crucial to understand both the spatial function, but also the cell surface proteins. So now I would like to introduce molecular pixelation to you, and the three-letter acronym there is MPX. So you will probably hear me saying MPX also throughout the presentation. So we're doing spatial proteomics for single cells, and we're looking at that in all three dimensions. And molecular pixelation uses DNA-encoded barcodes and next-generation sequencing to assess both abundance, but also spatial locations of the cell surface proteins. So what does the technology look like? So here we have an overview of the protocol, where the first step is to fix the cells. And fixation is carried out with PFA, and the reason behind that is to immobilize the cell surface proteome from the time of interest, so that nothing changes throughout the protocol. Then we have the antibody oligoconjugate binding, or AOC binding, with our 80-plex panel, which is basically barcoded antibodies that will bind to their target of the cell surface. Then there are two steps of pixelation, and this is where you add the spatial aspect. So pixelation A will create protein neighborhoods, and pixelation B connects these protein neighborhoods into one surface proteome map. And then there are a couple of steps of standard NGS library prep, like PCR and beads cleanup, before it's time for sequencing and later on data analysis. But I want to talk a little bit more of how this spatial dimension comes into play. So here we have a zoom in on a couple of the steps. And in the antibody binding, of course, you're adding the antibodies to bind to their markers. So at this step, you're retrieving the abundance information. So you get the surface protein abundance information, but at this point, we have no spatial connections. And in pixelation A, we add our reagent that's called the DNA pixels A. And that can bind to a couple of antibodies that are in proximity and give them a spatial tag. So all of these 4 antibodies got the same spatial tag, these 3 got the same spatial tag, and these 3. So you're getting these protein neighborhoods. And then we do pixelation B, which is basically another round of that, but it's going to bind in an overlapping fashion that then in silico can be used to connect the protein neighborhoods into one surface proteome map. So that is how you do with the technology, and then it's time for the sequencing. So sequencing with NGS on, for example, Illumina, and then you input your FASTQ files into Pixelator. That is the software that we have developed here at PixelGen, and it has 8 different stages. Stages for QC, filtering, but also for annotation and analysis. And there are 2 main outputs from Pixelator. So it's a web QC report that is, like its name, for quality control, assessing, for example, if it was sequenced deep enough. And then you have the other output, which is a pixel file, and that can be used for downstream analysis in ScanPy or Seurat to really tailor and answer your specific question in mind. So in the end there, you're able to get one unique spatial map of the surface proteome per cell. And using these, you can assess the protein abundance, the polarity score, and the co-localization score. And I'm going to talk more about that in the coming slides, what that is and how we use it. So we have generated a lot of documentation around the software. So just wanted to mention that here, to feel free to go into software.pixelgen.com and check out some software documentation, a lot of analysis tutorials to help guide the analysis, as well as some example data sets. And Pixelator is an open source project, so it's available to check out and download already now, and you can also find it in GitHub. Lastly, regarding the technology, so if you want to go deeper into the details there, our MPX paper has recently been published in Nature Methods. So you can either scan this QR code that I have here, or you can just find it online. And I just want to mention that it's an open access paper, so you can download it anytime you'd like. So let's talk a little bit about the molecular pixelation data. What do the results look like, and what is the data type of MPX? So I mentioned this previously, these three kind of hallmarks of molecular pixelation. So with MPX, you can assess protein abundance. You can start to look at protein polarity, as well as protein-protein co-localization. So when it comes to protein abundance, you can start to ask questions like, what is the relative abundance of each marker in my samples? What markers up or down regulate in my conditions? How much, to what extent? But also do more traditional UMAPs and cell annotation. And if you want to do immunomonitoring and see how frequencies change within populations, those are some of the things that you can do with the abundance. When it comes to polarity, which is one of our spatial metrics, well, if you look at this little picture here in the bottom, we have developed the polarity score to assess polarity. And it's telling you if the spatial distribution of this marker is non-random. So if it's centered around 0, that means that the protein is randomly dispersed on the cell surface. There is no typical pattern to it. Whereas if the polarity score is positive for that marker, it means that it's not random anymore. It is actually clustering to different poles or even polarizing to just one end of the cell. And then you can ask questions like how clustered or polarized is the marker? And also what markers increase or decrease in polarity in my conditions and to what extent? So the second spatial metric is the co-localization. And if we then start by looking at this figure again, we have the co-localization score that tells you, do these two markers co-localize more than we expect by chance? And if it's around 0, that again means that it's random, there's no pattern. But if it's negative, it means that these two proteins are actually segregating away from each other. And if it's positive, it means that the two proteins tend to be co-localizing on the cell surface. And then you can, of course, see how co-localized or segregated are them. Because we're realizing also with segregation, that seems to have a very big impact on biology as well when they're moving away from each other actively. So those are some of the hallmarks of molecular pixelation. Now I want to guide you through the MPX applications and some use case data. So the first use case is a drug mode of action. And here we looked at how molecular pixelation can be used to assess drug mode of action of the antibody drug rituximab. And I showed this figure in the beginning. I don't know if you remember, hope you do, but I will walk you through it again. So you have rituximab here in pink, and rituximab binds to CD20 on B cell cancers, which then polarizes CD20 on the B cell. And that makes it possible for NK cells to bind to initiate the ADCC pathway. So we figured this is a great example for molecular pixelation. And we took cells, and some of them were treated with rituximab, and some of them were not treated with rituximab. And then we checked under the microscope to see how it looked. And then you can see that for the control, it is randomly dispersed on the cell surface for CD20, whereas for the stimulated cells, it is actually polarizing to one end of the cell. So then we ran this through molecular pixelation, and we were able to see the same thing. So you can see here in blue, you have the negative control, whereas in red, you have rituximab treated. And if we start by looking at CD45, you can see that even with or without rituximab, it is centered around 0. And you can see that also here in this tessellation plot that it's randomly dispersed. Whereas for CD20, it's kind of centered around 0 for the negative control, but for the rituximab treated cells, you can see how it's increasing for the polarity score of CD20. So each dot here represents a cell that has a polarity score for then each marker, and here showing CD20. So molecular pixelation was clearly able to show the mode of action of rituximab. And then we have the second use case, and now I want to talk more of a basic biology kind of use case and T cell activation. So here we looked at how the spatial distribution of proteins and protein groups reveal biological insights that cannot be seen by protein abundance. This is actually an app note of ours. So I have the QR code here, but otherwise you can just go into the resource page of the web page if you want to read the full story on this one. But we started to just group them in a U map. So we have activated versus resting T cells. And then you see that you have the resting ones here in green to the right, and the stimulated ones in orange brown here to the left. And then we looked at CD82 and its abundance. We can see that there's not much of a difference here. The abundance is the same roughly between the stimulated and the non-stimulated groups. Whereas when you include the spatial aspect and you look at CD82 polarization, you see that it is very much polarized for the stimulated T cells, but not at all for the resting T cells. So there is something biologically that's happening when T cells become activated where CD82 plays a role. And we also have a 3D visualization tool that we have developed to look at the data from that perspective. And this is what it looked like when we used that one. So you have a resting T cell to the left, with the CD82 being colored here, and it's randomly dispersed. Whereas for the activated T cell, you can see how it's polarizing to several ends here. But of course, we want to validate everything that we do, so we use the confocal microscopy for this purpose. So then you have a resting T cell and CD82 colored in pink here. You can see how it's spreading out uniformly, no pattern to that. Whereas the activated T cell, you see that it's clearly clustering to several poles here. So it was nice to have that also validated with microscopy. But of course, that was protein polarity, and we also want to look at the co-localization. So what we're showing here in the heat map to the left, we're looking at a set of the proteins and how they are co-localizing. So this is then two different protein pairs, or one protein pair, two proteins. And then we found it interesting with CD55 and CD48. So you can see how CD55 is clearly increased in co-localization for the activated T cells. And we also included CD7 here just to show that it is not an increase in co-localization that's happening there. You can also start looking at it from this, more of looking at protein groups and more of a network of proteins in this co-localization network. And then you can see that CD55 and CD48 are co-localizing, but there are also several other proteins involved here. And we've decided not to show them so that other people can continue this exciting research. And as I mentioned, we want to validate this. But when it comes to this kind of co-localization, we needed super resolution microscopy. So we used super resolution microscopy, and that's what you see here. And we stained it with 3 different antibodies, so CD7, CD55, and CD48. CD7 is spreading uniformly on the cell surface, whereas CD55 and CD48 are forming nanoclusters on the membrane. And not only is it forming nanoclusters, but it's forming them on the same place. So we could confirm that CD55 and CD48 are indeed co-localizing. So very interesting. The third use case that I want to walk you through is a drug development use case. And here we looked at how abundance and spatial distribution can be used to assess compound effects of small molecules during the drug development process. This was actually a big pharma company who did this. And this is an overview of the process there, or what they had done before. So they had their drug development process, and they found two small molecules of interest that they wanted to know which of these compounds yields the biggest effect. Started off by looking at protein abundance. And you can see here how compound 1 is compared to the untreated samples, and compound 2 compared to untreated as well. And it's clear that compound 1 has a widespread effect, whereas compound 2 has some, but a smaller effect. Then we looked at protein polarization, and we could see the same thing. So here it's been specified for T cells, and you can see how compound 1 has bigger effects on polarity compared to compound 2. And here we've just zoomed in on one of the markers, where you can see how compound 2 and the untreated sample have very similar profiles centered around 0, whereas compound 1 has an increase in polarity. And then lastly, looking at protein-protein co-localization, we saw similar things, so more effects in compound 1 compared to compound 2. We wanted to zoom in to this marker pair right here, so co-localization of two different proteins. And same thing there, compound 2 and the untreated have very similar profiles, but compound 1 has an effect that makes them become co-localized. So overall, they found that that the compound 1 had bigger effects on all metrics, basically, which they validated in-house. And the quote that they told us after this was that this MPX study uncovered new important insights on the effects of the tested compounds. And that guided them to differentiate among candidates to move forward in their discovery program. So we were very happy that they could use it for that. So overall, there are a lot of different application examples where MPX has been used and also could be used. And what I've showed you here today with the drug development and pharma, looking at mode of action for different small molecules, but also antibody drugs like the rituximab case. We have also a lot of customers looking at hematology and doing blood cancer research. For example, with ALL and AML, and investigating treatment response. Immunotherapy and cell engineering is a big field for us. It seems like a lot of people are interested in looking at the CAR T effects, T cell exhaustion, and how long it takes for them to be exhausted, looking at checkpoint inhibitors, different treatment responses, doing time studies, very interesting seeing how things progress over time, both for the abundance, but also spatially, how things can become more and more polarized with time. And organoid models can be used as well. Also basic immunology, looking at T cell migration, differentiation, and what I showed you here today with T cell activation. So a lot of different applications. And I would like to show you, we didn't have time to look at that use case today, but here you have that, what I mentioned in the beginning, a T cell that's getting ready to migrate into a packed tumor environment, comparing to a control cell. And here it's been colored in blue for the markers that are polarizing when this migration event occurs. So this is the kind of 3D visualization that I have been mentioning that is possible to generate with molecular pixelation data. So lastly, just talking about the molecular pixelation product, you have it here clearly on the screen. So it's a reagent kit and a free software. And we have 80 protein markers for this sequencing-based spatial analysis of the cell surface proteome. And applications include the drug development, immunotherapy, basic biology, and hematology studies, like we talked about. And since it is a reagent kit, there is no specialized instrumentation or equipment. You can pipette. If you know how to pipette, basically, you will know how to do this. And it can be run in a standard lab with some centrifuge and PCR machine. Great. So with that, I'd like to thank you for your attention, and I welcome any questions. Thank you.
Fantastic. Thank you, Louise. And so, as mentioned, we do have some minutes now where we have an opportunity to answer some questions, and I will start with this one here. So, Louise, what level of bioinformatics competency is required to be able to analyze molecular pixelation data?
Yeah, right. That's a good question. So you do need some sort of bioinformatic knowledge and knowing how to get by in like ScanPy or Seurat. But there are a lot of material, like I mentioned, out there with tutorials to help guide. And we also have our support team to help out with everything.
Great. So another question here, what would be the advantage of running molecular pixelation opposed to, let's say, flow cytometry or mass spec, for example?
Right. Yeah. So with like we've been talking about now, so with molecular pixelation, you also get the spatial aspect, and you can look at a lot of different proteins at the same time. So you're again going to get this polarity metric and the co-localization metric for all of the different 80 protein markers. So you will be able to get the spatial metric, and compared to, for example, flow, all of the antibodies that are in the panel have also been extensively validated, work very well.
Great. And then can you combine molecular pixelation data with other omics data types, such as single-cell RNA-seq, site-seq, or tissue spatial data?
Yeah, great question. So what we have seen that customers have been doing so far, so like first of all, we don't have an omics platform to combine that yet, as I might add. But what we have seen that customers do is that they have a cell batch, and then they use some of the cells for example, single-cell RNA-seq, and some of the cells for molecular pixelation. And then you can compare the results from the different techniques. And of course, it's not going to be the exact same cell that you're comparing, but coming from the same biological sample. So it's a very interesting application, I think, to include that as well.
Yes. Great. And I think we'll have time for one more question. Does molecular pixelation work on other type of immune cells? We showed examples here for T cells, for example, but what about monocytes or NK cells and other immune cells? Yeah.
No, definitely. So the panel that we have in the product is a PBMC panel for human targets. So we have a lot of different, we have the backbone markers that you would need for the UMAPs, but we also have a lot of interesting targets for more checkpoint inhibitors and activation markers, etc. So all of the lineages that you find in PBMCs, we have them included in the panel. So like you mentioned, monocytes, B cells, NK cells, and so.
Mm-hmm. Great. So I think we'll continue. We do have more time at the end to answer more questions, but in the interest of time, we will now hear from Dr. Vyacheslav Milka and Dr. Vladimir Oleksiuk from BioLizard. And in the past 5 to 10 years, we've seen an incredible rate of innovation in the tissue spatial field, and the need to analyze complex data types and combine different omics data has increased immensely. So now I will let Slava and Vladi talk about some of the approaches and solutions that BioLizard and others are practicing. So the floor is yours, Slava.
All right, thank you for introduction, Annika. So good afternoon, everyone, from my side. In the next 20 minutes or so, I will be talking about data analysis of tissue-based spatial transcriptomics technologies. And of course, I will start with a brief introduction to the spatial omics field in general. So yeah, I guess now after nice examples Luisa gave, it goes without saying that knowledge on spatial location of biomolecules is super crucial for researchers to answer biological questions. So let's have a look on development of transcriptomics technologies. So around 15 or 20 years, if we take into account microarray, we've got a technology which allowed us to profile all genes in bulk samples. So that's beautiful RNA-seq technology. And then around 5 to 10 years ago, we reached the stage where we could profile the same number of genes, but in single cells. So we were able to separate different cell types. And finally, in recent years, we've got the beautiful technology group, which allows us to do comparable stuff, capturing of thousands of genes, but preserving their spatial location within tissue. So that's spatial RNA-seq. And that, of course, explains why these technologies are so popular nowadays. So let's have a look on the classification of spatial technologies. To the left, you can see two groups of spatial transcriptomics technologies. The first one is array-based, for example, Visium, StereoSeq. These technologies can capture whole transcriptomes, so they are untargeted. However, they, at least for now, they have lower resolution compared to second group, which I will be talking about a bit later. So the readout here is next generation sequencing. And the second group is image-based, or some call them in situ technologies. They have subcellular resolution, so they can capture individual transcripts within single cells. But they, at least again for now, they can capture only limited number of genes. So we are talking about hundreds or thousands of genes. And the result here is also next generation sequencing or fluorescence. If we look on this plot to the right, which profiles different spatial proteomics and transcriptomics technologies, taking into account the resolution and the number of markers, we can see that for now proteomics technologies still have lower complexity, so they can capture less proteins compared to transcriptomics technologies, which again might explain the popularity or fast adoption of spatial transcriptomics in recent years. I will give you an example of a data analysis workflow focusing on a concrete data set, at least for the vast majority of steps. And first, let's have a brief overview of the steps of data analysis. So actually, the first steps are very similar to the single-cell RNA-seq data analysis. So we start with gene cell matrix, which should be normalized, I mean, those counts. Then we usually find variable genes. Then we do dimensionality reduction, for example, principal component analysis. We can also do cell clustering of basis of those dimensions, and then we do cell annotation using gene markers. So actually, these steps are also performed in spatial transcriptomics. However, there are, of course, unique steps which can be performed only when spatial location of cells available. Actually, the cell segmentation tool is an upstream stage in analysis. This is very crucial. It's performed before gene cell count generation. The whole webinar can be dedicated to it, but since some technologies, or at least many commercial technologies, provide cell segmentation output, I will not focus on it in the next slides. However, we of course can argue about reliability and quality of those segmentations, because this is really a challenging step. While other steps are not provided and usually performed by researchers themselves, so I will be focusing on them. So obviously we can use location of cells to identify cellular neighborhoods within tissues. So I will be giving some examples of this. We can also perform dimensionality reduction, but this time taking into account location of cells, so spatial dimension. We can also apply some tools from single cell field to spatial. For example, trajectory analysis can be performed on those spatial niches or neighborhoods. And a big and important pillar of analysis is cell-cell communication. It's also something I will be showing you. So now let's have a look on the first obvious example of what we can do in the spatial field. So, but first, let's have a look on the examples of data sets for this. So here we will be talking about subcellular resolution technology. So genes belong to individual cells. The data set was generated by nanostring cosmics technology. So it's 1,000 genes targeted approach. The type of samples is human inflammatory samples. So we are talking about inflammatory bowel disease. So we have different conditions: ulcer, colitis, Crohn's disease, obviously healthy controls. And what adds complexity into the analysis of spatial is the presence of those number of different field of views from different tissues. So that's of course also taken into account in analysis. So spatial niche or neighborhood identification is kind of a logical first step in analysis. It can be seen as a city. If you travel to a new city, you probably want to attend areas of the city which are enriched with, I don't know, commercial buildings or sightseeing, and not industrial or purely residential buildings. So each region can be characterized by composition of particular buildings. So in the same way, tissues can be characterized. By compositions of different cell types, for example. And there are many algorithms by now how to do this cellular neighborhood identification. I like the one rather simple and elegant approach from the Gary Nolan group, where they slide tissues across tissues by using the cell windows of, for example, 100 neighboring cells. And then they compute composition of cell types within those windows. And then they repeat it for all cell windows within one tissue and do this for other field of use. So by this, they generate large matrix, which then can be clustered into, for example, 15 or 20 clusters, which are those spatial niches. And then you can see to the right lower corner example of 15 spatial niches on these 10 field of use. And these spatial niches then characterize both cell types and take into account location of cells within the tissue. This is powerful approach to characterize tissues. What we can do with them, with those spatial niches? Well, we can simply check composition of cell types within each niche. We can also profile and check inflammatory marker expression across niches, because in this case, for example, it's relevant because we study inflammation of patients. So I already mentioned before that we apply dimensionality reduction onto single cell dataset and spatial transcriptomics dataset. Actually, it's ironic that we researchers were striving for more genes, more samples in one experiment. And we reached the stage where the data became so complex that we need to apply this approaches to simplify again and to be able to answer biological questions. Luckily, there are those tools. For example, here a machine learning based package which performs non-negative matrix factorization can break down the matrix of this complex matrix. By the way, in this case, it's 500,000 cells, 1,000 genes. So it breaks down to a number of signatures or dimensions, you can call it, in this case, 10. And then what we can do, we can simply characterize each signature, checking them across the different cell types, or checking which signatures characterize conditions. So we can filter signatures and then kind of focus our data set to answer particular biological question. We can use those signatures to do a new clustering. So apply our favorite clustering and UMAP visualization, for example. And then we can apply trajectory analysis onto identified spatial niches. Why it is so powerful, I mean, this approach? Because we see our variable, our spatial niches as a continuum this time, not as a simply group of different spatial niches. So for example, we can find that a spatial niche can be located at the beginning of trajectory, which potentially or hypothetically characterizes our uninflamed samples. And then we can see how those spatial niches link to each other, how they go to a bifurcation point and diverge into, for example, different inflammation states like mild versus severe, or niches that characterize Crohn's disease versus ulcer colitis in our case. So we can really study those niches at this bifurcation point to understand what drives the pathology. And how do we do it exactly? We can pool genes associated with those points within trajectories. Again, complex bioinformatics tools can be used to do this. So in this case, negative binomial generalized model can predict or smooth expression of genes along each trajectory. So then we can say, okay, these genes are associated with this part of trajectory, so we are interested in it, and we can cluster them. So we can say, we can see which genes are upregulated at the end of those trajectories. And I remind you, those trajectories correspond to our spatial niches. So they do reflect the spatial architecture of the samples. And what we can do with those different cluster of genes, well, to answer the question, which pathways potentially involved in differentiation of pathology, we can do pathway enrichment analysis, either gene ontology or TGG. So that also can be extremely useful for researchers. Another tool or approach comparable to the previous one is gene set enrichment analysis. And here, the beauty of it is that we can say these pathways are enriched in this trajectory, and these pathways are enriched in the part of trajectory which is common for several trajectories. So that also helps us to understand how those trajectories, or in case of in our model pathologies, differentiate and progress. So I mentioned about the spatial niche identification. So we can identify 15, 20 spatial niches from cells, from 100, for example, neighboring cells. We can do it for 20 neighboring cells. I mean, the granularity can be different. And then how would we summarize all those spatial niches? Luckily, there are other beautiful tools like tensor decomposition analysis, where we can group enrichment of spatial niches across samples, taking into account replicates, and in this case, patients. And we can then see which spatial niches are enriched for example, inflamed samples, and to the left are spatial niches with granularity level 100 neighboring cells, and to the right are niches which were calculated from the granularity level of 20 neighboring cells. So we can really kind of zoom in on different granularity levels. Enough about spatial niches. Cell-cell interaction analysis is another very powerful tool, which we used also in single-cell RNA sequencing field. But there we were inferring those interactions. So we know ligand and receptor expression, but we don't know the location of cells, right? Because the tissues are homogenized. While here we do know location of cells within tissue because the architecture of tissue is preserved. However, we have less genes because in this case, we are talking about targeted approach. So, for example, it's 1,000 genes in this case. Nevertheless, we can check across conditions which cell types communicate between each other. And also we can zoom in into a particular pathway, right? Because we know which ligands and receptor pairs belong to each pathway, so we can select those and check which cell types communicate those ligand molecules, for example. And we can also do this for spatial niches instead of cell types. So that's, as I already said, a powerful tool to answer biological questions. Another way to visualize those matrices is to check these 2D plots where we can clearly see which cell types are enriched across different conditions, in this case, uninflamed versus inflamed. Before I mentioned, I was talking about the spatial transcriptomics technology with single cell resolution, and what to do if we use untargeted method which can capture whole transcriptome but lacks the single cell resolution. Well, luckily there are bioinformatics approaches to infer cell type composition within those spots or beads. By integrating these data with corresponding single-cell RNA-seq reference data set, ideally if it comes from the same tissue. Here I illustrate one of them, for example, cell2 location, where you can then, when you know the cell types within each spot, you can perform comparable analysis I just showed in previous slides. So that can be also performed. And another approach, which for some applications is also useful, is to find genes which have different expression profiles depending on the location within tissue. So so-called spatially variable genes. Different methods exist to detect them. For example, Moran's I method. And to the right, I added this stacked bar plot from a benchmark paper where they compare different tools to identify those variable genes, spatially variable genes. And the conclusion from there was striking to me. So the overlap between those methods is relatively low. So the unique genes, if you overlap all 8 methods they studied, is really a few percent for some technologies. For other technologies, like in this case was Visium or some other, it was still higher. So what I wanted to say by this is when you perform a spatial transcriptomics experiment and you want to do a good data analysis, the knowledge and expertise on all parts are extremely important. So obviously model used, biological model, then technology applied on it, and of course the tool set, so types of methods and tools to do the data analysis. So all parts are super crucial, as we can see from this small example, that results can vary depending on data, technology, and tool. And with this method, with this conclusion, I would like to finish my part of this talk and to give the word to a colleague of mine, Volodymyr, who will be talking briefly about another extremely important and also challenging part in data analysis, which is integration of different omics, so multi-omics analysis.
Yes, thank you. So we'll be stepping a little bit away from the spatial components, but don't worry, we'll dive right into them back. And I guess one of the key messages or purposes of this last part is to provide you with a little bit of the future perspective or vision and how spatial can help omics. So previously, you've heard about, well, molecular pixelation. Slava provided you with an overview of different methods and technologies within spatial transcriptomics and others. But actually, those are all different technologies trying to capture a small puzzle from, well, the whole biology on itself. And we can go beyond that concept, looking at, for example, flow cytometry, which was mentioned, but also MS-based proteomics, transcriptomics, and so forth. And the promise of multiomics is a little bit when you combine those different pieces of the puzzles, you will create a complete picture of biology, whereby you will have an increased understanding. So more than just combining those pieces of biologies, by combining those different omics, you would be able to get new insights that you will typically not be able to have when you're just looking at those different puzzles pieces. What is important as well is to really understand that, well, you have those different perspectives from those different technologies, but also each technology comes with its own biases, shortcomings, and its preferences. In a very simple example, when you're looking at, for example, bulk transcriptomics and bulk proteomics, even from similar samples, you will typically find a 40% difference in results or outputs or trends between both technologies, both as they capture different pieces of biology, they operate on a different level, but also from a technical perspective. Or a technological perspective. And this is just to kind of highlight the issues that arise when you are trying to combine different technologies with different gaps in them, and then you need to try to make sense from out of them. So a small introduction, but I would like to kind of briefly walk through a case study whereby we showcase some benefits of applying single cell level technologies to the multiomics, but we also envision a spatial component to further supplement, well, our multiomic solution. And to provide you with a little bit of context, so we collaborated over a larger period of time with a veterinary company focusing on, well, animal health. More specifically, they accumulated a lot of data for cats and dogs across different diseases. In this particular example, we focus on chronic kidney disease, and that data consists of transcriptomic, epigenetic, genetic, proteomic data from blood, tissues, but also imaging data accumulated across different projects in the period of years. So our first assignment was, of course, to kind of integrate all those different data resources and ensure that, well, we have a proper data management system in place, as this actually is the foundation for all the downstream analytical work. And then, well, we started out with the bulk approaches, integrate them into a multi-omic framework to get insights. One of the results there, and I'm cutting a little bit some corners here, is that, well, we did identify the usual markers, well, that are known from literature within our multi-omic approach, but we also identified some novel markers that brought some insights. Nevertheless, one of the issues was that, well, there was still a lot of variability within data, and while the majority of the results made a lot of sense, there were other parts that were puzzling researchers and the scientists. So within that context, we created a map of human CKD, whereby we combined multi-omic data on a bulk level, but also on a single cell level, to try and map it onto cats and dogs CKD data by learning a model on humans, and then use transfer learning to bring those insights onto cats, but still attain the specificity for, well, the specific species. And this actually brought some very interesting results, whereby we did see that part of the variability that we were seeing within the data that we could not place somewhere, and that, well, puzzled the scientist, can be attributed to, well, working with bulk samples. As on a single cell level, you have a much better granularity, and it allows you to understand where some signals come from. With a bulk sample, you have a mixture, as explained before. So this was one of the trigger points that kind of made us think, and where we now are experimenting with applying spatial technologies as well to bring another dimension of information and potentially unveil novel biology, or at least explain the results that we see within that context as well. So on the next slide, there is a brief overview of, or a high-level overview of kind of this data. There are a couple of challenges that I want to highlight here as well. The first one is obviously data integration. So certainly when you work with different omics, different technology, data integration is key. Not a topic that I want to focus upon right now, but another important challenge is actually to make sense from the data. So when you combine all those different data resources, you get so much valuable data that, well, you really need to leverage AI analytical tools to guide researchers and scientists to what is truly important and towards the most interesting trends within your data. And in my opinion, it's also within this context that AI and machine learning really shines. It's when you have highly dimensional, complex data, and you need assistance in order to make sense from the data. The last point is then, as a data scientist, you typically want to understand what is actually happening and don't want to take results for granted. So that's where visual tools enable us to kind of make sense from the data. We also develop biomics. Within that perspective, we saw a need for intuitively and easily being able to understand the data, to be able to question the data, not only from the AI perspective, but also from the biological perspective, enable scientists to look at the data, do some hypothesis testing, question the AI models, and so come to insights that can drive decisions. And those three challenges is also, or was, one of the drivers to create a new product, which is biomics, and to solve certain problems that we saw are particular for kind of those biological multi-omics data types, and well, we need a solution for. So on the last slide, there are a couple of conclusions just to summarize Slava's and my work. The first one is that spatial technologies can really bring an additional layer of information which can deliver new insight. Secondly, we need advanced analytics and AI tools to make sense from that data, to understand what is happening, and to kind of show us what is important within that data and distinguish it from noise. And thirdly, we really need to, well, with multi-omics, there is a new potential now with spatial to include it and, well, bring a better and a more complete picture of what is biological happening within different disease models. And with this, I would like to conclude, well, the presentation and receive questions for both Slava, myself, and PixelChin.
Fantastic. Thank you, Vladi and Slava. So important to be able to have tools to analyze your spatial data in this day and age. We do have a question from the attendee. So for the different omics platforms, how do you account for the difference in resolution? I don't know, Slava, if you want to try to answer that one.
So if the question for different omics, so meaning spatial proteomics or spatial transcriptomics, how we account for resolution? Well, you can see that different methods have different resolutions indeed. So you need to take this into account by checking if you have single cell resolution or not. If you don't have, then indeed you can apply those methods to kind of computationally bring it to single cell level. You can also compute those spatial niches by checking different granularity levels. In fact, you can mimic different resolutions by grouping neighboring cells, different number of neighboring cells, and that's how you can control resolution. So as I mentioned in example, 100 neighboring cells versus 20, you can do 300, you can do 10. or you simply work on single cell level, like in single cell RNA-seq experiment. I hope it answers. Not sure.
Great, thank you. And here's another question, I think, for you as well, Slava. Is it possible to integrate single-cell RNA-seq data set with spatial tissue transcriptomics data if they come from the same type of tissue?
Yes, absolutely. So that's actually even recommended, ideally to have from exactly the same tissue. So some people do even one slice goes to spatial, one slice goes to single-cell RNA-seq. And then this can be integrated and inferred either transfer of labels from cell types or other features. So yes, it's definitely possible. But nowadays, the technology evolves to the stage where we will have everything within one type of technology.
Great, thank you. So yes, we're at the top of the hour, and so we will circle back with the questions we didn't have time to answer live, so that you know. So yes, this concludes today's webinar, and I hope that we leave you with some new insights or ideas for your spatial work. And with that, we would like to thank you very much for joining us today. Bye-bye.